Hello students, welcome to EPG Pathshala. I am Dr. Hari Prasad, faculty at the Department of Biophysics, All India Institute of Medical Sciences. Today we are going to discuss about the module Two-Dimensional Electrophoresis under the paper Techniques in Molecular Biophysics Part 1. So students, let's see what we are going to learn in this particular module. We'll start by the introduction to 2D electrophoresis. Then we go on to methodology used in 2D electrophoresis, that would be pertaining to sample preparation, rehydration of the isoelectric strip, isoelectric focusing, SDSPAGE, staining, and then applications of two-dimensional electrophoresis, modifications of 2D electrophoresis, that is the differential in-gel expression or the DIGE, and finally some of the advantages and disadvantages of 2D electrophoresis will be discussed. 2D electrophoresis is a method of separating and identifying proteins in a sample by displacement in 2 dimensions, that is using the parameters of the isoelectric point and the molecular weight at right angles to one another. This could be used for analysis of complex protein mixtures extracted from cells, serum, tissues, or any other biological sample. So, what information would this give us? It will give us information pertaining to the PI of the protein, the molecular weight, and of course the quantitative expression, and if you are doing a comparative proteomics, the relative expression between two phenotypic conditions. Protein prefractionation. So the principle in two-dimensional electrophoresis comprises of subjecting proteins to two parameters, that is the isoelectric focusing and then the SDSPAGE electrophoresis. So after having done the 2D electrophoresis, we will have spots on that particular gel. Gels can be run off the sample and as well as the control group. And when we compare the differentially expressed proteins between these two groups, we identify certain proteins which have overexpression or relative underexpression. These spots are picked and then subjected to trypsin digestion. After that, they are identified using a mass spectrometric analysis. And having done or identified the sample, we then go for a validation because it's a population based study, and the validation could include experiments like Western blot, ELISA, immunohistochemistry, and cDNA expression profile. This way, we are in a position to either identify potential novel biomarkers or drug targets. So, what should be the ideal choice of a tissue for clinical proteomics? Then, for a researcher, tissue proteomics is always advantageous because he would then have to look for differentially expressed proteins within a few hundred proteins that are expressed at a tissue level. But it would be difficult for him to understand the differentially expressed protein in a systemic tissue such as either serum or plasma. Now, where does the doctor want to his biomarker to be in? A doctor has a feasibility of seeing the patient's serum or the plasma. So either at the OPD or at the ward, he would just draw a sample serum and then check for the presence or absence of a protein to know about the disease or response to treatment. So while the clinician wants it in the serum, it is feasible for the researcher to use tissue to start with a biomarker discovery. Therefore, in the bargain between the two, the ideal sample sometimes happens to be the fluid in the vicinity of that particular tissue. For example, if you take proteomic experiments of the brain, you probably could think of doing it on the CSF. If you are looking for biomarkers in lung, such as either lung cancer or tuberculosis, you have an option of looking at the protein expressions in the pleural effusion. If you are talking about cancers of the abdomen, there is an option of doing protein profiling on the ascitic fluid, and henceforth kidney diseases could be done on urine, and of course you could do experiments pertaining to tissues in and around the eye on the tear fluid. Complex nature of proteins requires a lot of sample preparation options. Recent developments of sample preparation and fractionation tools have increased the resolution of two-dimensional electrophoresis in clinical proteomics and also for biomarker discovery. Detailed laboratory methods and patented technologies are now available for sample preparation used to reduce the complexity of protein samples in biological tissues. Pre-fractionation uses an array of chemical reagents or antibodies, peptide libraries, chromatography, and electrophoresis methods to enrich the proteins of interest and deplete unwanted proteins, thereby increasing the signal-to-noise ratio in 2D electrophoresis. The technique of pre-fractionation depends on the experiment and the required outcomes. After the proteins have been solubilized, the next step involves pushing these protein solution into the IPG strip. The IPG strips are dry, therefore the process of subjecting them to the protein solution is called as rehydration of the strip. The strips are usually rehydrated with a 250 microliter of rehydration buffer that comprises of detergents such as 8 molar urea, 2 molar thiourea, 2% chaps, and DTT in a concentration of 0.003%. This particular rehydration solution is applied to the lanes on a tray that is called as the rehydration tray, and the IPG strips are overlaid on this particular lanes with the gel surface facing down so that they can absorb the protein solution into the gel. This particular strip is left overnight or probably for 14 hours time for the complete rehydration of the strip. The volume of the rehydration solution depends upon the length of the IPG strip and also on the amount of protein that is being taken up for the experiment. The rehydration tray, when it is left, it is usually overlaid with a specific type of oil, and the tray is covered to prevent any sort of evaporation to the atmospheric air. The first dimension in a 2D electrophoresis involves separation of proteins based upon the isoelectric point. What is isoelectric point? All proteins are made up of amino acids, and these amino acids have side chains, and some of these amino acids have certain amount of charge. The summation of all these charges on the amino acids constitutes the isoelectric point. And from an experimental point of view, isoelectric point is the pH of a solution in which the net charge of that particular protein is 0. So, in the first dimension, the proteins are subjected to electrophoresis on an IPG strip that is gradually increases in pH from one end to the other. So, when a protein is kept or is in a region of this particular gradient, there are certain amount of charges bestowed on this particular protein. Above its isoelectric point, a protein will have a net negative charge and will therefore migrate towards the anode. And below its isoelectric point, the protein on the IPG strip is positively charged and will migrate towards the cathode. As the migration takes place, the charge of them decreases, and finally when they reach a point where the net charge is 0, they continue to stay. This way, proteins, irrespective of where they are on the IPG strip, will focus to one particular point on the IPG strip, and therefore it is called as isoelectric focusing. The first dimension separates proteins now based upon their isoelectric charge. This is the first dimension. Proteins are subjected to isoelectric focusing. You need to have a gradient of pH between the two ends of a strip. So how is this particular gradient created for isoelectric focusing on the IPG strip? The gradient that is required is established by subjecting the solution of small molecules, what are called as polyampholytes, which have varying pi charges, to electrophoresis. Thereby, a pH gradient is now set up in the IPG strip. When proteins get into this particular IPG strip during the process of rehydration, the proteins in the varying pH environment will have different extent of charges as explained in the previous slide, and therefore they will have different rates of migration towards the anode or cathode, and that determines their focusing along that particular strip. So even as we have been understanding isoelectric focusing, what we now try to understand is, even as the protein migrates in the IPG strip, the overall charge on the protein will decrease until the protein reaches that particular region where the pH equals its pi, or in other words, the net charge on the protein is 0. Therefore, the proteins will now tend to focus into sharp stationary bands and will be positioned at a point in the pH gradient corresponding to its pi. Proteins that differ even by a single charge will now be fractionated and be separated into bands on the IPG strip as shown in the figure below. The technique of isoelectric focusing now involves removing the rehydrated strip from the rehydration tray and placing it on a particular holder called as the IEF holder. Wet paper strips will be placed at either ends of these strips, and it will be ensured that the strip is placed in a way that the gel faces upwards. Electrode clamps will be placed at the anode and the cathode, and this entire holder will now be covered with a glass cover. Now let's talk about the step called as the equilibration. Equilibration is a very important and integral step that is done after the isoelectric focusing but before the two-dimensional electrophoresis. What is it that is done during this particular step? The electrophorused isoelectric strip is first subjected to SDS and in the presence of dithiothreatol, and then it is exposed to SDS in the presence of iodoacetamide for 15 minutes each. What is it that happens in each step? Disulfide bridges that are reduced in the first stage of equilibration and the cysteine residues are permanently alkylated in the second stage. Therefore, proteins that are coming together at the end of isoelectric focusing and which have cysteines will now be prevented from forming disulfide bonds and thereby forming unwanted quaternary associations. The proteins in the second step will also be subjected to SDS, and therefore the proteins are coated by the SDS and therefore become negatively charged and ready for the electrophoresis in the next step, which is the SDSPAGE. The second dimension, or the SDSPAGE. After the equilibration step, the strips are now placed on acrylamide gels and subjected to electrophoresis. The principles and technique involve the same as that was explained in the module on SDSPAGE electrophoresis. The separation of these proteins will now be based on the mass to charge ratio. And as there is a negative charge SDS on all these molecules, the charge on the surface of the proteins are now compromised, and the negatively charged proteins will now go towards the positive end. After the completion of the SDS polyacrylamide gel electrophoresis, the gels are now taken up for staining. We have a number of options for the staining. For example, we have silver stain, the classical Comasi, and a modification of Comasi called as colloidal Comasi. Each technique has its own limits of sensitivity and detection. While silver stain can detect protein even to the extent of 1 nanogram, classical Comasi detects protein in 100 nanogram range, and colloidal Comasi detects protein in the concentration of 10 nanogram. The disadvantage with silver stain is it requires formaldehyde to develop the spots in the last step, and it is known that formaldehyde has a tendency to cross-link proteins. This affects its identification and the technique that is involved in its compatibility with mass spectrometry. So the reproducibility of colloidal Comasi is very high, and it is very low in the case of silver stain and classical Comasi. Now, we try to understand what are the applications of two-dimensional electrophoresis. Firstly, we have protein profiling. Snapshot representations of all the proteins in that particular phenotypic condition, organism or tissue can be known. Comparative proteomics, where protein profiles of two phenotypic conditions can be compared for biomarker discovery. This helps to know and understand differentially expressed proteins in different physiological and pathological states. Hundreds to thousands of polypeptides can be analyzed in one single run. Proteins can be separated in pure form from the resultant spots. The spots can be quantified and further analyzed by mass spectrometric analysis. Polypeptides can also be probed with antibodies and tested for post-translational modifications. It could also be used for differential expression of proteins between different cell types. Disadvantages: large amounts of sample handling, limited reproducibility, and smaller dynamic range than few of the other separational methods is one of the main concerns. It is also not automated for high throughput analysis. Certain proteins are difficult to run on a 2D page either because of the intrinsic hydrophobic nature of the protein, the low abundance, the extremely acidic and basic proteins cannot be got onto the particular gel, and also very large protein complexes or very small proteins can also not be resolved on the gels. Single protein can make multiple spots, therefore number of proteins could also be less than the number of spots. It could be either because of degradation, or it could also be because of multimeric associations. One last disadvantage of this particular technique is the experiment is as good as the staining technique, which dictates the sensitivity. Modifications of two-dimensional electrophoresis. This is what you call as differential in gel expression. 2D fluorescence difference gel electrophoresis uses a internal standard. So, the samples can be subjected to the same experimental conditions and can be easily compared and accurately quantified for different protein expression. DIGE also uses a technology of labeling proteins with cyanin fluorescent dyes. Also, importantly, it incorporates an internal standard, which is the pooling of all samples in an experiment and is run on every 2D dye gel in that particular experiment. Modifications of 2D. This is the dye where there is a standard for every spot on the gel and all gels within the same experiment are quantitatively linked. There is a gel to gel variations that are eliminated, making technical replicates optional. The internal standard is used to match and normalize the protein patterns across different gels, thereby negating the problem of inter-gel variation. Accurate quantification and accurate spot statistics between the gels can also be obtained for these experiments. We now try to understand the technique of differential in-gel electrophoresis that uses the technique of minimal labeling using cyanine dyes. 50 microgram of protein from either the control or the experiment is labeled with 400 picomoles of dye to ensure that the dye labeled is approximately 1 to 2% of the lysine dyes, so that the labeled proteins carry only one dye label and is therefore called as minimal labeling. Minimal dyes are intended for a general 2D application where sufficient amount of samples are available. For example, you could have a cyanine 2 label that is used for labeling controls, a cyanine 3 label used for the experimental group such as the cancer, and the Psi-5 that could be used as a internal standard, which would be labeling an equal amount of protein from all the samples in that particular experiment. The cyanine dyes, either the Psi-2, Psi-3 or the Psi-5 with different absorption and emission spectra, are basically N-hydroxy succinimidyl ester derivatives that are covalently tagged to the epsilon amino group of lysine residue of proteins. This replaces the epsilon amino group positive charge with the positive charge of the dye. The binding of the dyes introduce a small but matched increase in the molecular weight to the protein. Since these dyes are hydrophobic, they have a tendency to precipitate the proteins, and therefore only one lysine residue per protein is labeled, and therefore called as the minimal labeling. Minimal labeling limits the fluorescence intensity and thus the sensitivity of the stain, which could be taken as a disadvantage. The dyes are all charge matched and molecular mass matched to prevent alteration of the isoelectric point and to minimize the interference of the dye either to the isoelectric focusing technique or the SDSPAGE electrophoresis. The technique itself has an advantage of detecting proteins in the femtomolar range. Now, having understood about these cyanine dyes, we try to understand the advantages conferred by these side dyes on the dye technique. First of all, these cyanine dyes are spectrally very distinct, which means the cyanine 2, cyanine 3 and cyanine 5 dyes have discrete signals after the scanning with the typhon scanner. They are size and charge matched, and therefore the labeled samples will always co-migrate within the gel. That means to say, proteins coming from the experimental and the control group would be at the same spot. Each dye adds around 450 Dalton to the mass of the protein, which is not much to affect the SDSPAGE pattern. There is a possibility of multiplexing, which means protein labels with different of these cyanine dyed fluors will migrate to the same position on the SDSPAGE gel. As per the pH insensitivity, no changes in the signal will be observed over a wide pH. These cyanine dyes are photostable, and therefore there is a very good signal to loss noise ratio. The Sensitivity is in the range of picomolar to femtomolar range, which is otherwise not possible either with silver stain or Comasi stain. The technique of DIGE involves labeling 50 microgram of protein from an experimental group and labeling 50 microgram of protein from the control group. For example, you could be labeling the experimental and the control group with Psi3 and Psi5, and the SANIN 2 label could be used to label equal amount of protein from both these groups, which would now be the internal standard. Internal standard is used to normalize the expressions and also for in-gel comparison. After labeling, the proteins are mixed and subjected to isoelectric focusing, equilibration, and SDSPAGE. At the end of the SDSPAGE, the gel is now scanned with different wavelengths to create different images of the proteins labeled with the 3 different dyes. The images are now taken for analysis on a software called as the Decider software, where the relative quantification of each of the spots can be done. Spots with varying amount of differential expression are marked as differentially expressed proteins, and these spots are now taken, trypsin digested, and these peptide signatures are now used for identification using mass spectrometric analysis. The data generated on the mass spec machine is now used to match the theoretical mass to charge data that is available on the net, and the proteins are now identified. We have now understood the application of minimal labeling in DIGE technique. Let's go on to understand saturation labeling. Saturation labeling is designed to be used for applications where only small amounts of sample are available. The dyes here saturate the cysteine residues. The dyes are mass and charge matched, but there is a smaller chance of protein modification and isoelectric point shift on the isoelectric focusing technique. Saturating the cysteine dyes have a superior sensitivity as compared to minimal labeling, which is done on the lysine residues. As far as the sensitivity is concerned, even proteins less than 25 femtomole can be detected. However, the limitation of this technique is that proteins compulsory would require the presence of cysteine residues in their sequence. We now try to understand the advantages of the DIGE technique. Higher levels of detection is possible with a conventional staining of sensitivity in the range of nanogram per spot. There is higher accuracy with discrete signals and minimal crosstalk with higher accuracy. There are fewer number of gels required. There is a faster turnaround with the availability of sophisticated scanners, softwares, spot pickers, and the sophisticated mass spec instruments. It is reasonably cost effective considering the amount of data that could be generated. And last and the most important is that all the proteins from any of the samples are subjected to the same experimental conditions. Even though we see colorful pictures and data from the DIGE technique, there are certain problems that are encountered with the gel-based proteomics. They are: extensive workforce and consumables are needed, presence of high abundant proteins camouflages the expression and the visibility of low abundant proteins, The sensitivity of detection is sometimes not good for these low abundant proteins such as the cytokines. There's absence of protein in the 2D gel due to extremes of pH and the molecular weight. There is sometimes difficulty to reproduce the results, and this complicates biological variation and the experimental variations. There's difficulty to maintain homogeneity between the samples of the same group. For example, if we are considering samples of tuberculosis, it would be very difficult to have samples of per se only the tuberculosis. You could always have this particular condition along with other coexisting conditions like pneumonia or viral cold. There is a specific need to validate the gel-based experiments with other supportive experiments such as Western blots, ELISA, cDNA profiling, and immunohistochemistry as part of the validation. There are sometimes contaminants like carbohydrate, lipid, and ionic impurities, including salt, that could affect a smooth run of a gel. Variations in two-dimensional gel electrophoresis is sometimes a problem. The problem could arise because of system-related variations, inherent biological variations, or induced biological variations. Just have a glance at some of the aspects of these variations. System-related variations. These variations that occur from gel to gel are either researcher induced in terms of pipetting. They could be because of different conditions of isoelectric focusing and the 2D electrophoretic technique, or it could also be due to certain types of editing and interpretation while using sophisticated software at the time of data analysis. Next, the biological variations. Inherent variations occur from sample to sample and organism to organism, and this induces a lot of confusion on the aspect of expression of certain proteins in certain conditions across phenotype of organisms or clinical samples. These places the onus on the clinicians to collect homogeneous samples or to have patients with a homogeneous profile before categorizing them into any of the physiological or pathological phenotypes. And lastly, biological variations could also be induced by the clinician or the researcher by subjecting patients to a particular drug regime. It could be a lifestyle change on part of the patient, or there could be differences due to the disease state. So students, we now come to the end of this particular module. So let's summarize what we have learned so far in this particular module. 2D electrophoresis is an experiment used for the analysis of complex protein mixtures extracted from cells, serum, tissues, or any other biological samples, including the patient samples. 2D electrophoresis involves solubilization of the proteins, reducing the non-protein contamination, isoelectric focusing on the IPG strips, SDSPAGE electrophoresis, and staining for the visualization of these protein spots. Applications of 2D electrophoresis includes protein profiling and differential expression analysis for drug target identification and biomarker discovery. DIGE is an improved modification of the 2D electrophoresis which allows for qualitative and relative quantification of proteins between different phenotypic isolates by incorporating the internal standard. Advantages and disadvantages of 2D electrophoresis are there in this particular technique and have to be factored in depending upon the availability of protein, sensitivity of the mass spectrometric machine, the cost, and most importantly, the availability of tissue samples. To an extent, non-gel based proteomics has been able to offset some of these disadvantages that are seen in gel based proteomics. Thank you so much.